Hello, good evening. Good evening, people. I hope you can hear me. Hello, hello. How are you? People, our plan is to discuss today the paper, not the paper, sorry, this tute. So find the tute. This one. Find it. In the class, we discuss some other questions. Right. People, we had to discuss questions today. Right. So, where did we stop in the theory class? How many questions we discussed? As I remember, we discussed last Saturday. Last Saturday, we discussed nucleus. Did we discuss nucleus? Can someone chat with me? Did we discuss nucleus last? Page number 9. What are the... Santosh says 23. Okay, 23. We discussed 23, ne? 23rd question. So, take the tute. Take the tute. We'll go to page number 9. Page number 9. Page number 9. Is it page 9? Page 10, ne? Page 10. Page 10. Right. We had to discuss the 23rd question. Nucleus. Nucleus. Right. Can we... shall we discuss question number 23? Yes, Res. Yes, Res. Right. Nucleus, question number 23. Nucleus is a light microscopic structure. What am I remote? Nucleus is a light microscopic structure. Brightness එක වැඩි පේන්නේ. මේක සෙට් කරලා නැහනේ. නෑ මේක සෙට් කරලා නැහැ. Right. Question number. මේකේ pen ටික කො. Right. Question number 23. Nucleus is a light microscopic structure which is about 5 micrometers in diameter. Label all components of the following diagram. Number 1, nuclear pore. We labeled it last time, I can remember. Nuclear pore. Number 2, what's number 2? Number 2, nuclear envelope. Number 2, nuclear envelope. Number 3, we'll say nucleolus. Number 3, nucleolus. Number 4, we'll take people nuclear plasm. Nuclear plasm. Number 4, not very clear. Number 5 and 6, we said people chromatin. 5 and 6, we say chromatin. 5 and 6 together, we'll say chromatin, but 5 and 6 need not to label it separately because 5 is heterochromatin and 6 is euchromatin. You can learn it later, not now, no need. Right, question number 24. What is the composition of chromatin? What are you going to write, people? Composition of chromatin? DNA and histone protein are right there. DNA and histone protein. DNA and histone protein. DNA and histone protein. All right. Question number 25. Chromatin becomes chromosomes. Chromatin becomes chromosomes. Question number 25. Chromatin become chromosomes during cell division. Describe how this condensation happens. You have to describe how this condensation take place. Now you have to say that people, upon forming the chromosome, chromatin further condenses with histone protein. Right? Right there. Chromatin further condense with histone protein. Right? You don't need to know any particular process. Harida. Chromatin further condenses with histone protein and transform into chromosomes and transforms into chromosomes. Right. Question number 26. Question number 26. Nucleolus is a spherical structure. Nucleolus is a spherical structure. State the function of the nucleolus. What did you learn, people? What is the function of the nucleolus? Right? Yeah, okay, 24 doesn't matter. Right? What is the function of the nucleolus? Right? This is involving in formation of ribosomal RNA, right? Ribosomal RNA and ribosomal subunits. We'll take it as involving in forming ribosomal RNA and ribosomal subunits, right? The nucleolus. We said nucleolus synthesizing ribosomal RNA and ribosomal subunits required for protein synthesis. Okay, moving to next question, people. Moving to next question, question number 27. What are functions carried out by nucleus? Functions of the nucleus. Now you can write a list of functions. What do you write, people? Control all cellular activities. Control all cellular activities. Control all cellular activities. Synthesize DNA to produce new nuclei. Synthesize DNA to produce new nuclei for cell division. Synthesize ribosomal RNA and ribosomal subunits. You can take that also as a function of the nucleus. Synthesize mRNA and tRNA. So answer to 25 once again. Okay, wait. Synthesize mRNA and tRNA. Store and transport genetic information. Store and transport Store and transport genetic information. Store and transport genetic information. Right. Next question, people. Next question. Question number 28. Give structural differences between nucleus and nucleolus. Nucleus and nucleolus. Give structural difference between nucleus and nucleolus. What are you going to say, people? What are you going to say? Now, nucleus is membrane-bounded structure, right? What else you can say? Nucleus contain DNA covered with histone protein. That means the DNA is not naked, right? DNA is not naked. How about in the nucleolus? Sorry. How about in the nucleoid? Right? How about in the nucleoid? Circular DNA present and not covered by histone protein. Right? Okay. Not covered by histone protein. I will write that, people. Nucleus is membrane bounded organelle. Right there. Nucleus is membrane bounded organelle. Nucleus is membrane bounded organelle with DNA associated with histone protein. With DNA associated with histone protein. Associated with histone protein. Right? Nucleoid found in prokaryotes. Nucleoid found in prokaryotes with naked DNA. With naked DNA not associated with histone protein. Not associated with histone protein. Right. Moving to question number 29. Moving to question number 29. Moving to question number 29. Ribosomes are organelles involving in protein synthesis. Check that. Ribosomes are organelles. Make it maru in the bani. Ribosomes are organelles involving in protein synthesis. Make an in table in. Ribosomes are organelles involving in protein synthesis. Briefly describe how they are organized. Consist of two subunits. They consist of two subunits. Consist of or made of rRNA and proteins. Made of rRNA and proteins. Made of rRNA and proteins. Na yasra sargi na eka. Harigi na bene kakya nevi. Me hatara dennu ko dala harita. Teen lara kain karannu ko den galalan tine. Consist of two subunits. Consist of ribosomal RNA and proteins. All right. Right. Moving to question number 30. What are free ribosomes and bound ribosomes? What are free ribosomes and bound ribosomes? What are you going to say, people? Huh? Free ribosomes are the ribosomes found in the. What are free ribosomes and bound ribosomes? Ribosomes available freely in the cytoplasm are free ribosomes. Ribosomes freely available in the cytoplasm are free ribosomes. They are free ribosomes. And bound ribosomes are present attached to membrane surface of RER. Bound ribosomes are present attached to membrane surface of RER. Membrane surface of RER. Membrane surface of RER. Right, moving to next question, people. What are two types of endoplasmic reticulum? Actually, reticula, neda. Plural word of reticulum, reticula. Right, what are two types of endoplasmic reticula or reticulum? Doesn't matter. Ne, SCR and RER. Right, moving to people, next question. Question number 32. Compare functions of rough endoplasmic reticulum and smooth endoplasmic reticulum. Compare functions of rough endoplasmic reticulum and smooth endoplasmic reticulum. What are you going to write for that? We know, people, some functional differences are there. Functional similarities available too. Functional similarities present. Functional differences are present too. Ne? What is a functional similarity? Both are involving in forming transport vesicles. SCR and RER both are involving in forming transport vesicle. SCR and RER, they asked this question last year, people. 2024. A-level paper, they asked that. But similarities and dissimilarities in SCR and RER. That's the last question of this tute, right? So, two similarities are there, ne? Right? Two similarities. What is that? Synthesis of phospholipids is also common for both. RER synthesize membrane phospholipids, SCR synthesize phospholipids too, right? So, compare functions of rough endoplasmic reticulum and smooth endoplasmic reticulum. So, we'll write compare and contrast. Similarities and dissimilarities. Just say compare, the comparison. We'll write that right there. The both SCR and RER involved in forming transport vesicles. Both SCR and RER involving in forming transport vesicles and phospholipids involving in forming transport vesicles and phospholipids. We'll take it as a similarity. Then you can say, right, RER involved in forming glycoproteins. RER synthesize glycoproteins. SCR synthesize lipids, steroids, and oils. SCR synthesize oils, steroids, lipids including oil, steroids, and phospholipids. SCR synthesize oils and steroids. Oils and steroids. RER does not involve in detoxification. RER does not involve in detoxification and calcium storage, but SCR carries out detoxification and calcium storage. Right? I think writing that sort of an answer is going to be sufficient. Right, people, we are moving to next question. Question number 33. Question number 33. Right? Question number 33. 34, ne? Right, 33. 33, they ask, what is the connection between ER, Golgi and lysosome? Can you tell me the connection between ER? You know, ER makes vesicles, ne? ER makes vesicles. Those ER vesicles become the ER vesicles become Golgi. Golgi is forming vesicles too. Golgi forms vesicles too. Right? What happened with the Golgi vesicles? Golgi vesicles are forming lysosomes. Golgi vesicles form lysosomes. So we discussed that this is ER, right? They are ER, we'll take that SCR and RER, ne? SCR and RER. This is the rough one. So they make vesicles, they form vesicles. You know, these vesicles are carrying people. These vesicles carry phospholipids, the glycolipids, glycoproteins, phospholipids, steroids, and those stuff. So they come and build up what? Golgi cisternate. They build up Golgi cisternate. So this is Golgi. This is Golgi cisternate. And then Golgi forms vesicles. Golgi makes vesicles. These Golgi vesicles, some of them transforms into lysosomes. Some of the Golgi vesicles, they transforms into lysosomes. They transforms into lysosomes, right? So therefore they are ER. This is Golgi, and then lysosomes, right? So there is a connection. So we'll write that, people. ER vesicles form Golgi apparatus. ER vesicles form Golgi apparatus. And Golgi vesicles form lysosomes. Golgi vesicles form lysosomes. Golgi vesicles form lysosomes. People, can you send your chat to the main chat group? Because I can't see all the chats now. Okay, I can't read the chats. Right. Question 33, what is the problem? So can you repeat that question 33? ER vesicles form Golgi apparatus. Golgi vesicles form lysosomes. That is all. Lysosomes. Right. So can't we label nuclear matrix instead of nucleoplasm? You know, you can take nuclear matrix and nucleoplasm. Yeah, okay, no issue. But nuclear matrix is, you have to know the difference. It's some sort of a protein network inside, right? So it's not visible when you just show it to the matrix, right? When you show it to the matrix, that's it, right? Okay. The next one, question number 34. State main functions of Golgi complex. State main functions of Golgi complex. You will ask a lot of questions. Sir, will you upload the recording to the website? Normally, all BioEve ones are coming under our advanced online free videos. Now, when you go to the advanced online website, there is a part called as the free videos. So, biology free videos, chemistry free videos, physics free videos, right? In that category, all past these evening sessions are going to be there, right? So you can watch them later, and then I don't have the ability to upload the tute now. I can't do it now, right? I'll do it later if you request. I have to do multitasking here. That's my problem. So people, maybe for the newcomers, you can join with the proper class. I can give you the PDF, but it's now in several different computers. I can't handle it here, right? So I'll give it for the newcomers. We have a Monday class. We discuss these theory parts from the beginning. Now today we had the class, we are still discussing microscope and then cell structure and all. So if you're interested, you can join on Monday, right? So you can learn the theory part plus you can, this is just some question discussion work. All right. So question number 34, state main functions of Golgi complex. Main functions of Golgi complex. Question number 34. Main functions of Golgi complex. Main functions of Golgi complex. Right? So, main functions of Golgi complex. We can say collecting, modifying, packaging, collecting, modifying, packaging, and distribution of materials. Collecting, modifying, packaging, and distribution of materials. Second one, manufacturing cellulose and non-cellulose components of cell walls such as pectin. Now you have to pay attention. Cellulose and non-cellulose components such as pectin. Now they have not mentioned hemicellulose, right? So we have to go strictly with the resource book. Cellulose and pectin they have mentioned. Other non-cellulose materials, all right? Cellulose and non-cellulose components such as pectin, production of lysosomes. Production of lysosomes. Right, moving to next question, set people. Lysosomes. Lysosomes. Lysosomes contain hydrolytic enzyme. Lysosomes contain hydrolytic enzyme. State those hydrolytic enzymes. Lysosomes contain hydrolytic enzymes. State those hydrolytic enzymes. Now, people in the resource book, they have not mentioned specifically the names of these hydrolytic enzymes. So, we say that carbohydrate breakdown enzymes, protein breakdown enzymes, lipid and nucleic acid breakdown enzymes. We'll take the answer like that, right? What are they, people? They are the carbohydrate, protein, lipid, and nucleic acid breakdown enzymes. We can say nucleases, carbohydrases, lipases, and proteases, right? But we'll go according to the resource book statement, right? They are enzymes break down carbohydrates, lipids, nucleic acids, and proteins. Nucleic acids and proteins. All right. So then this diagram is showing you people some of the events of the lysosome. Some of the functions of the lysosomes are given here in this diagram. Now you have to label 1, 2, 3. 1, 2, 3 people. 1, 2, and 3. 1, 2, and 3. So they ask what these 1, 2, 3. You can see that people, the materials which come from outside, they are digested with the help of lysosomes. So what you can say the first function, people? Digestion of food particles received by endocytosis. Digestion of food particles. Digestion of food particles received by endocytosis. That's number one. Number one. Second one, people, autolysis causing cell death. Now, what happened here? An old organelle. What is this, people? Old organelle. Old organelle. Old cell organelles like peroxisomes, like mitochondria, they can be removed with the help of the lysosomes. We call this as, people, autolysis. What do you call it? Number two, autolysis. That is autolysis. So, mitochondria and other old cell organelles are removed by lysosomes by a process called as autolysis. And then next one, people, number three, this is, people, transport after the digestion, removal of any residue materials. Removal of any residue materials. Removal of any residue materials, right, is transport of residues. So we'll take that third function, transport of residue materials. transport of residue materials. That is done by lysosomes, transport of residue materials. Is that clear? Right? So question number 35. Now we move to question number 36. Question number 36. What are different functions of peroxisomes? What are different functions of peroxisomes? What are different functions of peroxisomes? Well, number one, detoxification of peroxides. You know, peroxisomes are involved in breakdown of hydrogen peroxide. They break down hydrogen peroxide. You learn people functions of peroxides, breakdown of hydrogen peroxide, H2O2 into water. So H2O2 into water, right? H2O2 broken down by what on that peroxisomes. They store people catalase, ne? Catalase enzyme. This catalase enzyme break down hydrogen peroxide, which is produced during the metabolic activities of the plant cells or animal cells. You learn some cells like leaf cells, storage tissues. They produce hydrogen peroxide. And also you learn people, you also learn liver cells. Chicken liver, I showed you a video in the class also. Chicken liver, you can try people putting some hydrogen peroxide to piece of chicken liver and it forms people a lot of oxygen, right? So this breakdown hydrogen peroxide into water plus oxygen. You have to balance this equation, 2H2O2, 2 water. I think now it's balanced, right? So catalase enzyme functions. So we'll write there, people. What are the different functions of peroxisomes? Detoxification of peroxides. First one, detoxification of peroxide. Number two, photorespiration in plants. I'll explain photorespiration later, right? In photosynthesis lesson, we'll be discussing, right? There is a special process happen in plants, right? During daytime, which is somewhat similar to cellular respiration, right? We call that as photorespiration, right? Photorespiration, peroxisomes involving in that. Then a special modified peroxisome called as glycosomes. Special modified peroxisome, what do you call that? Glycosomes are involved in conversion of fatty acids into sugars. Convert fatty acids into sugars. So three functions we discussed, my dear. So write that. Question number 37, the last structured question. Neither last structured question. What are glycosomes? State importance of it. I mentioned it now, right? These are modified form of peroxisomes which are involving in conversion of fatty acids into sugars, right? Specialized peroxisome found in fat stored tissues that convert fatty acid into sugars, that converts fatty acids into sugars. All right. Okay. Right. We move now to MCQs. MCQs, people, MCQs. Right. Which of the following cannot be seen under light microscope? Which of the following cannot be seen under light microscope? All right. Which of the following cannot be seen under light microscope? The starch grains. Can you see starch grains under light microscope? People, starch grains, you can see them present inside some chloroplast, right? Sometimes people, some of the plastids which involving in storage. Did you hear something called the leucoplast? Did you learn anything called leucoplast? I told you, there are three types of plastids: chromoplast, leucoplast, chloroplast. So starch grains can be stored in these plastids and they are visible to the light microscope also. Cannot be seen under light microscope. It can't be starch grain. You can see the starch grain under the light microscope. How about the yeast cell, people? Do you think the answer is yeast cell? Can you see a yeast cell under light microscope? Yes, you can see yeast cells. So yeast cells are not going to be the answer. Plasmid. Now these plasmids are structures present inside bacteria. Ne? So they are structures present inside bacteria, and there is no way for you to see a plasmid because even a bacteria cell is not clearly visible. Ne? Even a bacterial cell is not clearly visible, right? So therefore, starch grain is wrong, yeast cell is wrong, plasmid is correct, chloroplast can't be, stroma of the chloroplast cannot be. Ne? So one of the past paper question, right? You found the answer now. Answer is the third one. Question number 2. Which of the following common in plant, animal and bacterial cells? Which of the following is common in plant, animal and bacterial cell? Can you type me the answer, people? All these common one is the ribosome, right? Ribosome must be the answer. Ribosome is the answer, right, people? Next one. Next one, question number 3. Which of the following statement is not included in the cell theory? Look at these statements very closely. Look at the statements, people. Right? Which of the following all organisms are composed of one or more cells? You learn it. The basic structural unit is the cell. You learn it. Cell is a basic functional unit. Learn that. All cells arise from pre-existing cells. Learn it. Who proposed that? Who said that all cells arise from pre-existing cells? Do you know the name of that person? Right? Was that you? Did you say that? Eh? Santosh says Virchow, Rudolf Virchow. That's good. Rudolf Virchow. That is Namidu. Namidu. That's good. Namidu. Right. So all cells are microscopic. We didn't learn that. All cells are microscopic. Is that true, people? Can't all cells be microscopic? Are there any big cells which are not microscopic that you can see to your eye? Aren't all cells are microscopic? What do you say about that, people? All cells are microscopic? Eh? Eh? All cells are microscopic. Lamai, do you know any cell which is not microscopic, visible to your naked eye? That's good. Yes. Mariam says yes, Lu. Namidu says yes, Lu. What is that, people? Yes, that is the great the Humpty Dumpty, Humpty Dumpty cell. That is the egg cell, the egg yolk, of course, right? Not the whole egg, egg yolk, that the yellowish color spot. Yeah, so that's not microscopic. I said that the sailor's eyeball, right? There is a large algal cell, people, right? Large cell, which is clearly visible to the eye. So some exceptions are there. So some exceptions are there. Question number 4, people. Moving to question number 4. Which of the following statements regarding microscope is correct? Which of the following statement regarding microscope is correct? In the light microscope, the visible light passed through the objective lens and then through the specimen. Is that correct? Light is passing through the objective lens and then through the specimen. Is that correct, people? Is that correct? Light is passing through the objective lens, and then the people passing through the specimen. You know the light microscope arrangement, neither. So you know this upper lens is the eyepiece lens. Then the objective lenses are fit here. This is the objective lens set is the another. Objective lens set present. And then here you have the stage, people. And on the stage you keep the specimen. Right? Beneath the stage, people, there is a lens present. Do you know the name of the lens that present beneath the stage? Do you know the name of the lens which is present beneath the stage? What do you call that lens, people? What do you call that lens? This one I ask, what do you call that lens? Condenser. Condenser lens. And here you have the specimen. Specimen is that. And here you have the objective lens. And then here you have the eyepiece. Eyepiece. There you have the eyepiece. So therefore they say now light which passes through. Now light is actually reflected on the mirror, passing through the condenser lens, passes through the condenser lens, it passes through the condenser lens, light comes like that, passes through the condenser lens, then pass through the specimen, then enter into the objective lens, and then passes up people to your eye. So therefore, this statement is not correct. Neither. This statement is not correct. The statement is not correct. Why, people? It says the light microscope, visible light passes through the objective lens and then through the specimen. Then through the specimen. So that is wrong. Question number 2, people, statement 2, projection of a light beam through the vacuum is the principle of electron microscope. Projection of the light beam. What is this? Light beam in electron microscope. People, in electron microscope, light beam. Neither. So that's wrong. Neither. Wrong statement. Scanning electron microscope is used to study internal structure of the cell. Either correct, scanning electron microscope to internal structure, or it is the surface details. What do you say about that, people? Yes, you can type me. Yes, surface details, so that's wrong. Transmission electron microscope is used for detailed studies of living specimens. Living? Living? Oh, can you see living specimens? You can't see any living specimens. Why you can't see any living specimens in electron microscope? You stain the cell with heavy metals which are toxic. Right? Not that only. You put this guy, you dry him until it loses entire water. Because you can't keep a wet specimen, people, in the electron microscope. You cannot put a wet specimen. Right? So you have to dry it. Next thing is, you put this guy in a vacuum. Electron microscope has a vacuum. So there is no way that you can observe, people, any of the live specimen. That is not correct. No live specimen, my dear. So that is wrong. Magnification and the resolution powers are important properties of microscopes. Answer is 5. Answer is 5. Right, moving to question number 5. Which of the following may be seen in both prokaryotic and eukaryotic cells? People, which of the following may be seen in prokaryotic and eukaryotic cell? Nuclear membrane, no. Cytoskeleton, no. Cell wall, yes. Peptidoglycan, cell wall in prokaryotes. Eukaryotes are having cell wall with cellulose or maybe chitin neither. And then people, flagella, correct. You learn prokaryotic flagella. Prokaryote flagella, what's the diameter? I can't remember the diameter of the prokaryotic flagella, my dear. Do you know the diameter of the prokaryotic flagellum and the eukaryotic flagellum? Do you know the diameter difference? Rima says 20 nanometers and 20 nanometers. That is prokaryotic. And then eukaryotic, what is the diameter of the 200? Rumana says, Santosh and Rima, 200 kilometers. Kilometers. Oh, nanometers. Nanometers, neither. And then can you tell me another difference between the prokaryotic flagellum and the eukaryotic flagellum? Can you see me another difference between those two, people? Can you tell me another difference? Other than these, can you tell me another difference between them? Intracellular and extracellular. Very good. That's Rima. Intracellular and extracellular. But I need another difference, not this difference. Can you tell me another difference other than this one? One of you says, Navidu says 9 plus 2 microtubule arrangement in eukaryotic cells. That is correct. Perfect answer. Yeah, microtubules are present in the eukaryotic one, and then you don't see that in the prokaryotic one. That's good. Very good. I have some bright people in the class, right? They're shining, ne? They shine in this meeting, right? Mariam says prokaryotes lack microtubules and eukaryotes. That's correct. That's correct. Right? Intracellular, extracellular. So you said all the required points. That's good. Very good. So we have C and D as the answer. C and D is the answer. So we move further down. And I'm taking people question number 6. Question number 6. Have a close look. Question number 6. Generally, the highest magnification and the resolution power of the compound light microscope are respectively. What's the answer? Highest magnification and the resolution power of the compound light microscopes are respectively. What is your answer for that? Can you tell me the answer number? Sir, couldn't hear you properly. First one, low. Someone says fourth. Is it fourth? 1500.2 millimeter. Fourth one can't be, no? So it has to be 1200 nanometers. Answer is the first one. Question number 7. Steps involved in observing an onion epidermal peel mounted on a glass slide and placed on the stage of the compound light microscope are as follows. Steps involving in observing onion peel. Onion peel. You're going to observe. And then, what is the procedure? What is the procedure? Adjust the mirror. Use fine focus. Ayyo. What is the order? ACB, neither. ACB, second one. That's too simple, ne? ACB, second one is the answer. Right, people? Question number 8 is yours. Some features of cells are as follows. Presence of plasma membrane, presence of 70S ribosomes, occurrence of mitosis, presence of subcellular components suspended in cytosol. Which of the above features are common to prokaryotes and eukaryotic cells? Presence of plasma membrane present in all. Presence of 70S ribosomes. That you can take neither. 70S ribosomes. What this is, some features which are the above common to prokaryotic and eukaryotic cells. 70S ribosomes are present inside the chloroplast and mitochondria in eukaryotes. So we'll take that 70S ribosomes present. Occurrence of mitosis, no. Prokaryotes undergo binary fission, so you can't take mitosis. That is wrong. And presence of subcellular components suspended in the cytosol. Subcellular components suspended in the cytosol. What do you say about that? I'm taking it as correct. So my answers are A, B, D. I'm taking A, B, D. Do you agree with me? Is it A, B, D? A, B, and D. A, B, D only. ABD 2020, ABD, huh? ABD, 8th question, ne? ABD 5, ne? ABD 5. ABD 5. Right, people? ABD 5. Question number 9. Now, question number 9. 2021 A-level paper question. 2021 A-level paper question. Right. Which of the following statements regarding plasma membrane is correct? Mainly composed of carbohydrates, phospholipids, and proteins. Mainly composed of carbohydrate. Oh, that's wrong. Phospholipids and proteins, fine. Phospholipid molecules are movable and provide fluid nature to the membrane. Yeah. Peripheral proteins are tightly attached to the outer surface of the membrane. Phospholipid bilayer enables the nearby cells to communicate with each other, enabling nearby cells to communicate by phospholipids. Now, actually, what helps the cells to communicate? What actually helps the nearby cells to communicate with each other? Is it by the phospholipids? Do they talk with each other through the phospholipids, people? It's like you talk with somebody with your ear, with your fingers. Can you talk with your fingers? Maybe, sometimes, if you pinch and all. So, this is actually done by the proteins. Hydrophobic tails of phospholipids attached to cytoskeletal fibers. What's that, people? Right, this is A-level exam, the exam question. Hydrophilic tails of phospholipids are attached into the cytoskeletal fibers. That's wrong, people. That's wrong. So we can't take that. So we got the answer anyway. Answer is number 2. So answer is number 2. Right, moving to next question, people. Question number 10. In compound light microscope, 22 A-level paper. Look at this. A-level 2022. A-level 2022. Lens reflect the light to magnify. Reflecting or refracting? What's that, people? Lens reflect to magnify the image of the specimen observed. Not reflect, neither. Wrong. Refract. Resolution power is inversely proportional. Resolution power is inversely proportional to the wavelength of the light. Resolution power is inversely proportional to the wavelength. Now, wavelength decreases, inversely proportional resolution power. Wavelength of light. Resolution power is inversely proportional to the wavelength of light. Lesser the wavelength, greater the resolution power. Right? That is correct. That is correct. You learn this equation, neither. Resolution equal resolution is equal, what you said, people? Wavelength divided by 2 kilo. So if you take a light beam, minimum wavelength of light beam is 400 nanometers. 400 nanometers divided by 2 is 200 nanometers. So that we took as the resolution of the light microscope. So second is okay. The image produced by the eyepiece lens is magnified by the objective lens. Image produced by the eyepiece lens, is that possible? Eyepiece lens image is now magnified. Look at this. Then the light has to come from your eye down to the specimen. If the light, if you are like, if your eye emits light, you know that in some cartoons, some people are there, they emit laser beams from the eye. If you are that sort of a guy, then you can send the light first through the eyepiece lens, then you can send the light through the objective lens, and then you can send it through the specimen, and you can burn the specimen. Then you can burn the specimen. So actually how it should happen, people? from the mirror, condenser, then through the specimen, then the objective. Objective lens image is further magnified by the eyepiece. So it has to be like that. It has to be like that. So here we cannot agree with this statement. The image produced by the eyepiece, wrong. The maximum magnification is usually 600 times the actual size of the specimen. No, 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 no. How many times? Maximum magnification, people, 1,000 times. Why do you say 600 times? 1,000 times. Resolution power is zero 0.2 millimeters. Resolution power is not 0 0.2 millimeters. Resolution power is 200 nanometers. 200 nanometers, my dear. 200 nanometers. Ne? 200 nanometers. So that's wrong. Right, moving to next question. Question about nucleus. Question about nucleus. Now, we have a lot of questions. Don't worry. A lot of questions. We are moving people to the structured essay paper. Page number 20. Past paper questions. We'll just keep this for a short time. We'll move for some of the structured questions. Let's move to some of the structured questions, right? We'll keep some questions. We'll keep some MCQs to discuss in the next class, right? It's better, people, we discuss them with a delay. I'll be starting you a series of papers, but not now, right? We'll take a little more time because we have at least a couple of lessons. At least we had to do unit 2 to keep a test, right? So then you will be getting people some regular papers, right? Maybe one paper in 2 weeks time, right? So we'll discuss the past theory parts right here. In 1 week, some revision of quick revision on the theory. Next week, people, we will go on discussing the questions relevant to that. That is what we do with the 26 people. That's their project, right? So we'll start when time to come. We'll start it for you also, right? So until that, people, we'll go on discussing these tutorial questions, some extra questions that I give you in the class. We'll take this time every Monday. I don't like Monday, but anyway, now no alternative, right? So we'll take some extra work on Monday, right? Maybe 1.5 hours maximum. You don't have to stay for another more than another 20 minutes, right? We'll be finishing, so don't worry, right? Hang on, hang on, hang on, right? So A level is all about, right, hanging, right, holding. You have to have some strong mentality, ne? To bear up, tolerate the difficulty. Sometimes you might think that this time period is too sleepy, too difficult, phone is difficult, my phone is small, and then you can say so many things, right? Right? You can say so many excuses you can give, but that's not useful, right? Just do it. Neither. Do it. Somehow do it. Somehow do it. Right? No point of giving excuses for A levels, right? So you are going to be rewarded for your hard work, right? This is a training. A level is a training course. This is like army training, right? You train well, then you are rewarded. Neither. So this is a life training, not just to go to university, right? To work throughout your life properly, right? So question number 2001 structured essay paper question. The question 1 and 2 are based on the diagrams of the electron micrograph of the part of the animal cell given below. So this is animal cell, part of the animal cell. They want you to label it. That's a very simple task. A level examination, they give you such a diagram and then ask you to label people. See how easy A level. What is this G? Can somebody tell me what G is? right? I don't need in order, but can someone say what is G? You can see it's like sugar bun neda. It's like sugar bun. Seeni bunnies wage neda lamai. Oh, hori bunnies. You can see a lot of neda tiny dots are there. What is that? That's RER. That's good. RER. But you write the full term. Rough, rough, endoplasmic, endoplasmic, rough endoplasmic reticulum. Right? In the A-level examination, no short things, huh? No short things. You write, don't write RER, just write rough endoplasmic reticulum. Some years they have not given marks for writing RER, right? Ilangada F, what is that, people? What is that? That is the outermost part, smooth part. Then that is smooth endoplasmic, smooth endoplasmic, endoplasmic, smooth endoplasmic reticulum, smooth endoplasmic reticulum. Then people, I can't actually identify H. Can you help me to identify H? What is H? Can somebody? Very good. Golgi apparatus. Golgi apparatus. Golgi apparatus. Very good. And then people, what is E? Do you know what E is? That's the nucleus. Very good. Nucleus. That is nucleus. Very nice. Nucleus. So that's your task. That's your A-level task. Name the structures labeled as E, F, G, H. What are major functions of G and H? What is G? That is RER. What is H? That is Golgi. So RER functions, you can mention RER functions, people. Transport protein synthesized by bound ribosomes, production of transport vesicles, synthesizing glycoproteins. Anyone you can write here. Anyone. Golgi. Golgi, what you can say? Formation of lysosome. That's easy, ne? Golgi forming lysosomes, collecting, packaging, modifying, distribution of materials, manufacturing cellulose, non-cellulose materials, correct. So you can write them here. Question C1. What are four major types of organic compounds found in eukaryotic cells? Four major organic compounds in eukaryotic cells. That's not a question, Neil. What are those four people? Proteins, nucleic acids, lipids, and carbohydrates. Carbohydrates. Right, moving to next page, people. State the major polymeric compounds found in following. State the major polymeric compounds found in following. Plant cell wall. Plant cell wall, what is the major polymeric compound? You can say cellulose. You can say cellulose, pectin, hemicellulose, hemicellulose. Bacterial cell wall. What is the material? Peptidoglycan. Peptidoglycan. And then what is the storage material in the liver cells? What is that, people? Glycogen. And then ribosomes, polymeric material, ribosomal RNA, and protein. And protein. And then what is the common respiratory substrate in living cells? Common respiratory substrate in living cells is glucose. That is glucose. Describe the simple test to confirm the presence of reducing sugars in germinating mung seeds. Germinating mung seeds. Simple test. Now, this is not a cell structure function question. This question actually comes from, which one, people? This question comes from the chemical basis of life lesson. So what is the test that you have, right? What is the test that you can do to say that the mung seeds are having reducing sugars? Describe the simple test. Now you can't just write Failings test or the Benedict test. Now Failings not in our syllabus. We discuss only Benedicts, right? So we'll use the Benedicts here. So what are you going to say, people? Prepare, right? Mung seed extract. Can you start writing? You right now. You're too sleepy. Get up, get up. Right. Now, right there. Some people are even eating roti, me. Pain, roti kana, me. Classic, right? So, I can see some videos are turned on, right? And so, Lamai, one of the Kenny first say people prepare green gram seed extract. Alianda, prepare mung seed extract. Prepare extract of mung seedlings. How is it? Then take 2 milliliters into a test tube. Take 2 milliliters into a test tube. Add equal volume of Benedict solution. 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 How is it? Then heat it in a boiling water bath. Heat that in a boiling water bath. Right? Heat that in a boiling water bath. Right? If brick red precipitate forms, if brick red precipitate forms, that indicates presence of reducing sugar. That indicates the presence of reducing sugar. That indicates the presence of reducing sugars. That indicates the presence of reducing sugars. That's it. That's it. You can show it easily. Right? You can show that easily. All right. You can show that easily. Right. Moving people to next question, 2013. Old syllabus question. What is cytoskeleton? What is cytoskeleton? Supporting structure of the cell which maintain its shape. Can you write that? Supporting structure of the cell which maintains its shape. That is cytoskeleton. Supporting structure of the cell which maintain its shape. What are main components of cytoskeleton? What are main components of cytoskeleton, people? Microtubule, actin filaments, right there. Microtubules, actin filaments, microtubules, microtubules, microfilaments, microfilaments, and what is the third one? Intermediate filaments, ne? Intermediate filaments. State 4 functions of cytoskeleton other than providing support. 4 functions of cytoskeleton other than providing support. What are you going to write? What are you going to write? Functions of cytoskeleton, people. Provide strength to cytoplasm. Provide strength to cytoplasm. Maintain shape of the cell. Good. Movement of cytoplasm. Movement of cytoplasm. Encourage of organelles and cytoskeletal enzymes. Yeah, good. That's good. Santosh. 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 What's the meaning of that word Santosh? Happy? Eh? Santosh means happy. Happiness, ne? Santosh. Happiness. Happiest, ne? Santosh is the happiest in the class. Santosai. In singular word, ne? Singular word, ne? Right? Santosai. Santosai. Ahanda Santosai. Santosh. Bhatiya and Santosh, ne? Bhatiya Santosh. Right? So, Santosh and the others, that's enough for the day, ne? It's enough for the day. We'll meet once again, people, next Monday. We'll try to make this time period more productive, right? I need your support, and I need your support. I'm very sure that this 2027 batch, right, is going to help me, right, to do this task, right? So we'll meet on every Monday. We'll fix it. 8 p.m. is okay, ne? 8 p.m. okay, ne, guys? 8 p.m. okay, ne? This time is all right, ne? 1 hour and 15 minutes, right? We can do something the mid of the week, right? I'll do something productive. Don't worry. I'll help you to revise the sections, right? When you accumulate your workload, people, I'm here. I'm here. Mehuna, right? Mehuna, right? In Hindi say ne, Mehuna. Mehuna innava, right? To help you to revise your sections, right? So we'll do that. So thank you. Salute for all of you for joining with the class tonight. And then people expecting you to see soon, Saturday. Bye-bye. See you.